So I thought I'd run through the mitosis test that I set you and you've done, because there are some harder questions on there, and even if you see the mark scheme, you aren't necessarily going to fully appreciate what's going on. So if we start off by looking at question 1, you've got a diagram of the cell cycle, and then you've got some information in this table. You've got the different stages, or you're looking at the nucleus within a cell. You've then got the number of chromosomes and just a mass of DNA which relates to those chromosomes. So I've just drawn a diagram to help, hopefully, explain what the answers are. So you're told that in prophase there are 26 chromosomes with a mass of 60 units. What I'm not going to do is draw out 26 chromosomes because that'll take forever. So let's say that here in prophase we have got the number of chromosomes is 2, but I'm still going to use the same mass of DNA. So that's 60 units. You've then got to, with the question, is complete the number of chromosomes and the mass of DNA for telophase, and then within a sperm. And really, this is an application question, getting you to, do you understand what's happening in each of the steps of mitosis? So let's start off with prophase. We know that in prophase, the cell has had to have gone through interphase, and crucially, semi-conservative replication has occurred. So here's the cell, here's the nucleus in interphase, early interphase, and so semi-conservative replication hasn't taken place. So this species has got a diploid number of 2 chromosomes, a homologous pair. Semi-conservative replication takes place. We have still got, this is the crucial bit, we have still got 1, 2 chromosomes, but in this case each chromosome consists of a sister chromatid. So in prophase we need to know that semi-conservative replication has occurred. So we've got 2 chromosomes consisting of 2 sister chromatids with a mass of 60. Then we go through metaphase, anaphase, and so crucially we need to, again, but we have to understand in anaphase, spindle fibers are going to contract, the centromere is going to split, and now the chromosome breaks apart. The 2 chromatids now start to move to opposite poles. So then when we get to telophase, I suppose I should stick in my arrows just set. When we get to telophase, we've still got in each of the nuclei of the cells, we've still got, in terms of the number of chromosomes, 2 chromosomes, as in 1, 2, 1, 2. But here's the tricky bit, and it is tricky. When we start to talk about the mass of DNA in one of these nuclei, because each chromosome is just one DNA molecule wound around histone, the mass is going to be half of what it was in prophase, because each chromosome consisted of 2 chromatids. So the mass has to be half. The mass has to be 30. And that's difficult, and that's tricky. It's a hard question. When we then look at a sperm, as it says in the table, so I just put gamete down here, we need to know that meiosis cell division has taken place, and you're going to have, within a sperm, within a gamete, you're going to have half the chromosome number, so that's going to be 1. And in terms of the mass, well, the mass has to be half of what you would find within a cell of this species. So we know you've got 2 chromosomes in interphase, and so it's just the same as you would have in telophase once that separation has taken place. So if a cell of this type has got 2 chromosomes with a mass of 30, then the gamete must have 1 chromosome with half the mass. That's tricky, that's a difficult one. In question B, the key to this is understanding what mutation in a gene leads to in terms of the protein. So it says if the DNA of the cell is damaged, a protein called p53 stops the cell cycle. Mutation in the gene for p53 can cause cancer to develop. You've got to convey the idea that if there's a mutation in the gene, the p53 protein, you could say it's either not made or synthesized, or you could say it's non-functional. That's the key there. You're not expected to know specific drugs that treat cancer, and the key to this is it says at what phase in the cell cycle would each of the drugs act. So a drug that prevents DNA replication, you need to know that replication occurs in interphase. A drug that prevents spindle fibers from shortening, shortening takes place in anaphase, pulling the chromatids to opposite poles. In question number 2, you just need to know the method for the root tip squash. In B, you're being asked to calculate, again, key here, calculate the percentage of the cells in which the chromosomes are visible and would consist of a pair of chromatids joined together. So chromosomes aren't visible in interphase. Prophase, metaphase, anaphase, telophase, the chromosomes are visible. When do they consist of a pair of chromatids joined together? Or as we've seen just here, in prophase, chromatids joined together, and metaphase, chromatids joined together, but anaphase, telophase, the chromatids are split apart. So in terms of what you're doing there, you're adding prophase and metaphase together. You're then dividing by the total, which is 982, and then times 100 you get a percentage, 9.4%. For B part 2, when you're being asked on another occasion with a different garlic root tip, why do you get a different set of results? We're dealing with living things, we're dealing with organisms, and as we've said before, there's going to be genetic variation within those organisms, different garlic types. The other factors then that are going to affect the rate of mitosis are other environmental factors. So the age of the root tip, water availability, temperature, nutrient availability, things like that. Question 3a, I'm not going to say anything about that, you just need to run through how DNA is replicated. B is tougher. The graph shows information about the movement of chromatids in a cell that has just started metaphase of mitosis. Alright, so we've got start of metaphase at 0, and we've got time on the x-axis in minutes, and we've got distance on the y-axis. So initially to answer the question, to start off with, all you can use is this dotty line, which is the distance between the chromatids. Right, here's one chromosome, metaphase, and in metaphase the chromatids are joined via a centromere. Here's our chromosome. So we're looking at each chromatid. Here's the start of metaphase at 0, and you can see that time elapses and the distance between the chromatids is 0. Therefore, that's telling you that the chromatids are still together effectively within that chromosome. You need to sort out your scale. Each of the 2 little blocks is 1 minute. So we've got 15, 16, 17. At 18 seconds, the distance between the chromatids starts to increase. It increases, increases, increases. Well, the only reason why the distance between the chromatids is going to increase is because anaphase starts, and each of those chromatids from the chromosome starts to get pulled to opposite poles. At then 25, 26, 27, at 28 minutes onwards, the distance between the chromatids remains constant, so that's inferring then that anaphase has stopped at that point. So B part 1, what was the duration of metaphase in this cell? It starts at 0, and it's the point at which the distance between the chromatids starts to increase, because as soon as the chromatids move apart, then anaphase starts. So that's 18 minutes. The second question is use line X to calculate the duration of anaphase in this cell. Well, we know that anaphase starts when the chromatids' distance starts to increase between them. So it starts at 18 minutes. The distance between the chromatids increases until 28 minutes. So the difference between 18 and 28 is 10 minutes. The third question, complete line Y on the graph. So line Y, crucially, is the distance between each chromatid and the pole to which it is moving. Okay, the distance between each chromatid and the pole it's moving. Well, we know that anaphase starts at 18 minutes. So to start off with, the distance between the chromatid and the pole that they're moving to is going to be constant. So the distance there is 20 micrometers. So that is going to remain constant until anaphase takes place and starts to decrease the distance between the chromatid and the pole it's moving to. So again, we know that anaphase starts at 18 minutes. So initially you're going to have a constant distance up to 18 minutes, and then the distance between each chromatid and the pole to which it's moving to is going to decrease down to 0, where the chromatid reaches the pole it's moving to. C, you're looking at the percentage of cells in each stage of a cell cycle in two different tissues. One's cancerous, the other isn't, and then you've got the stage of each of the cell cycles. In tissue sample C, one cell cycle took 24 hours. So we know that this took 24 hours. Use the data in the table to calculate the time in which these cells were in interphase. So look, we can see that interphase spends 82% of the whole cell cycle. You've been told that the whole cell cycle takes 24 hours, so it's 82% of 24 hours. So multiply 0.82 by 24 and you get 19.68 or 19.7 hours. Now, I know that we usually talk about hours and minutes, but you'll accept that as an answer. And that was pretty much it.